Good afternoon and welcome once again to Cowen & Company's 39th Annual Healthcare Conference. I'm Phil Nadeau, one of the biotech analysts here at Cowen, and it's my pleasure to introduce the next presenting company, Editas. Those of you who heard our session at 8 o'clock this morning with three luminaries, in particular George Church, know how exciting CRISPR is and the whole idea of gene editing. So really happy to have with us today Charlie Albright, the CSO of Editas. There'll be a half-hour presentation in this room and then a breakout immediately following in the extra room, which is right next door. Charlie.
Thank you. Well, thank you for the nice introduction and welcome to spring in Boston. Developing medicines with an innovative technology to make new transformational medicines is an area with incredible promise, and it's also obviously an area with some risk. And so we'll be making a lot of forward-looking statements today and encourage you in your leisure to read the fine print. Our mission is to use the incredible power of CRISPR gene editing to develop innovative and transformational medicines. And today I want to update you on our progress in those endeavors. And 2018 was a significant year for us. We got our IND approved at the end of the year, approved in 30 days, which is the first ever CRISPR in vivo CRISPR gene editing. We're poised to recruit patients in the middle of this year for that trial and dose patients in the second half of the year, and we're super excited about that. LCA10, the lead indication there, not only being the first in vivo CRISPR medicine, but also sets us up for subsequent in vivo CRISPR medicines. And I'll talk more about the USH2A program, another target for inherited retinal diseases, and it benefits tremendously from the LCA10 program. And further, the LCA10 program begins to set us up to think about in vivo editing in other tissues. And so as I'll talk, we're uniquely in a position to initiate those activities compared to the other editing platforms and feel that that'll be a very rich area of investigation as the years go on. We're also making tremendous progress in the second pillar of our strategy, which is ex vivo editing. Our sickle cell program, I'll go through some of the details about that. We have reason to believe we have a best-in-class therapy there. We showed some of that data at ASH last year, and I'll review some of that as well. As you can expect to see some exciting data in that area this year. And so this puts us in a good position to execute against our EM22 goals, which are to have 3 compounds in early development, 2 compounds either in or poised for late development, and built on the backbone of a best-in-class platform, culture, and technology. And so we'll give examples of all of those in the presentation today. So there are 2 pillars to our portfolio strategy. On the left side, I show the ex vivo editing, which is our lead program there is LCA10, a subretinally delivered AAV for Leber congenital amaurosis type 10. We are doing these in the ophthalmology program in collaboration with Allergan, and I'll go through more details there. And as I said a minute ago, this really sets us up to be able to do in vivo editing in other tissues. The other pillar of our portfolio strategy is really ex vivo or editing outside the body. Sickle cell disease is our lead program there. I'll talk quite a bit about that. We also feel that cell-based medicines and oncology are a tremendous opportunity, and we're really excited about both of those. But let me start with the ophthalmology portfolio. We started in ophthalmology because for several reasons. First, there are thousands of patients with severe diseases where we really understand the biology of those diseases. And that lets us go directly at the cause of those diseases. The pathway to both clinical and regulatory success is well worked out. So you'll see in the LCA10 program we're delivering the modality in exactly the same way that the approved drug from Spark is delivered. And so we're building very much on the backbone of the gene therapy work that's gone in the ocular diseases, of course, one of the most successful areas for gene therapy at this point in time. Our lead program is for LCA10, an inherited form of blindness. The children are essentially born blind with this disease or severely visually impaired. This is a collaboration with Allergan. It's a 50-50 co-development and co-commercialization in the United States. And the collaboration is going quite well. A little bit about the disease then. So LCA10 is caused by a mutation in a protein known as CEP290. CEP290 is involved in making the outer segments, which are shown in red on in this cartoon. And the outer segments are part of the photoreceptors shown in blue, and it's those outer segments that actually absorb the light, which leads to the vision. And those outer segments in you and I are turned over about every 2 weeks. And so without the CEP290 protein, you see our version of what a shriveled set of outer segments look like. What we hope to do is actually regrow those outer segments to make them look like normal outer segments. And if we do that, we believe we should be able to, those patients should be able to sense light and hopefully use that sensation of light to create a vision. The LCA10 genotype that we're tracking is due to a point mutation in an intron. We're going to use two guides to cut out that portion of the DNA that encodes that point mutation, thereby enabling normal splicing to happen, because that point mutation causes cryptic splicing. So this is part of how the understanding of the disease really allows us to design a molecular therapy directed directly at the cause of that disease. We've disclosed a ton of data on this program, and that data was recently summarized in a paper that was accepted in Nature Medicine. I just want to show you two tidbits of that data here that pertain to the clinical aspects. Shown on this slide is the rate of editing versus time in weeks here in a mouse. So we've done studies in mice, and the studies in non-human primates are quite similar to this. And this shows that the editing is complete by about 6 weeks. And so this, combined with our non-human primate data, reads directly into what we expect to see in the clinic, because we expect to see similar kinetics of editing in humans. And so that editing is then going to lead to the correction of that mutation I just described on the last slide, regrowth of the outer segments, and hopefully restoration of vision. So the bottom line is we expect to see changes in a small number of months following administration of the drug in this study. Equally important with AAV therapies or any therapy is tolerability, and shown on this slide are 4 different measures of tolerability in the non-human primate. You can see that all measures are 0 to 1, which means the drug was well tolerated by all 4 of these measures, and in fact the differences between the placebo and the drug are not meaningful in this. And this just shows that the drug, when administered subretinally, like the Spark drug is well tolerated in non-human primates. That, combined with our dose projections, which again are very close to where the therapeutic doses are for Spark, give us confidence that we can deliver the editing machinery via an AAV to humans at a dose that will be well tolerated. And this, combined with a whole lot of other data, went into the IND package and has enabled us to get the IND approved by the FDA in 30 days, which we consider a significant accomplishment. In addition to the preclinical work, we are conducting a natural history study. The natural history study does several things for us. It's a study where we're going to look at about 40 patients to look at endpoints and start sites. Those are going to then enable the clinical trial design that's shown here. That's a fairly typical phase 1, phase 2 study for a gene therapy for ocular diseases. It's going to be a dose escalation study. We're going to start with patients that are adults that have light perception only, which means they can only distinguish light and dark. They have no perceptible vision. That's obviously the safest place to start for a new therapy. We're then going to dose escalate to doses that will achieve what we believe will be the maximal amount of editing. We'll also progress into patients that have some amount of vision. Eventually, we will progress then into children that have various levels of vision, because a priori we expect that children with some level of vision are the most likely to benefit from a therapy of this kind. We're obviously starting in patients where the risk-benefit is highly favorable with a new and innovative drug. Our second program in the inherited retinal space is for Usher syndrome type 2A, another form of inherited blindness. It will benefit tremendously from the LCA10 program. In particular, we're going to use the same vector, the same promoter, and so within the AAV, there will really only be 40 nucleotides that differ out of the 5 kb that's going to be packaged in the AAV. We'll also benefit from the pharmacology models we set up for LCA10, the specificity and safety models we set up for LCA10, and the IND package will have many, many similarities to that for LCA10. And this is going to be a theme in all of the ophthalmology programs and is part of what's gone into our building the portfolio. We're doing this as a collaboration with Eric Pierce at the Massachusetts Eye and Ear, and we've disclosed some of the data establishing proof of concept last year at the American Society of Gene and Cell Therapy, and we'll disclose additional data further validating our approach at this year's ASGCT meeting in May or April. The second pillar of our portfolio strategy is ex vivo, or cell-based medicines. We believe that cell-based medicines represent an entirely new class, a new modality in the drug discovery arena. We further believe that essentially every cell-based medicine is going to be made better by gene editing. And therefore, it's an important place for us to play for those reasons and because our technology works incredibly well ex vivo. And so our programs at this point are focused on editing hematopoietic stem cells for beta hemoglobinopathies and on cell types for oncology, which include engineered T cells and NK cells. Let's start with our medicine for sickle cell disease. This is obviously an area of tremendous unmet need. It's also an area where we really understand both the biology and the clinical aspects of the disease. So in particular, we know from humans that those that have elevated levels of fetal hemoglobin can suppress the symptoms of the mutant allele that's present in sickle cell disease. So we're trying to recapitulate that human genetics by elevating the level of fetal hemoglobin. And so when we go after this, we'd like to see several valuable attributes in the product. We obviously need to be able to successfully edit ex vivo the long-term progenitors that are part of the population of CD34 cells that we're editing. We need to see that those cells will implant and maintain and repopulate, particularly the erythroid lineage, which is what we're trying to correct in these patients, thereby leading to a durable therapy, not a short-term therapy. And so we've gone at this by targeting the beta globin locus. That's different than what our competitors have done, which is to target an enhancer of the BCL11A gene. BCL11 is a negative regulator of fetal hemoglobin expression. We've decided to go after the region of the chromosome that actually contains the mutations that lead to elevated fetal hemoglobin in humans. So we screened a 300 kilobase region of the genome with 26,000 guide RNAs, and I've identified many sites that increase fetal hemoglobin, including those that have been identified by human genetics. And so we found the best of those sites, and we've taken some of those into a relatively new animal model, and we've shown, not surprisingly, that when you edit the beta globin locus, you're able to repopulate the erythroid lineage just as well as cells that have been mock edited, which is shown on the left, and the rates of cell death or apoptosis are no different than the controls. We were surprised to find that when you edited the BCL11A enhancer, you actually see a dramatically decreased level of erythroid progenitors, and that's shown on the far right-hand side of this slide. And that is a result of increased cell death or apoptosis. And so we were able to see this because we used a new mouse model that actually allowed us to measure the erythroid lineage, and that wasn't possible in the preceding mouse models that have been used to conduct these experiments. So none of these results conflict with the published data. We presented this story at ASH last year, and we were selected as one of 11 of the best of ASH posters out of about 5,000 abstracts. So we were gratified by that, and that reaffirmed our work. As well, we've shown that our approach leads to elevated fetal hemoglobin levels, both by when we look at the cells ex vivo, as shown on the left-hand side, and when we look at the cells that are put back into those mice. So I have more to say about this as the year goes on. Suffice it to say that we also think we'll be differentiated based on the amount of fetal hemoglobin we'll induce, and we'll show more of that data in the coming year. In addition to the program targeting sickle cell disease, we have a variety of programs targeting cells for oncology. We have a collaboration with Juno, now Celgene, for engineered T cells in oncology. We've achieved a variety of technical milestones there, and we'll have more to say about that as the year goes on. We're also very interested in NK cells. These are a cell type that are outside of the Celgene collaboration, and we believe hold great promise as cell-based medicines to treat both liquid and solid tumors. These technical programs are really built on the backbone of an incredible platform, which is also a significant basis of differentiation for what we do. They're the broadest suite of enzymes. The Staph aureus Cas9 that is part of all of the in vivo CRISPR medicines that we're making is uniquely licensed to us. The Strep pyogenes Cas9, which everyone else uses, is too large to be packaged in a single AAV, so we are uniquely able to do in vivo editing, and that's a substantial advantage. As well, the CPF enzyme is turning out to be an incredibly good enzyme. We'll have more to say about that in the background of the sickle cell program later this year, but it allows us to do things that we would not have been able to do with Cas9. In addition, the IP for the CPF1 stands independent of the Cas9 IP. We've built an ability to deliver with AAV, which stands unique in this field. Our ability to characterize on and off target editing is among the best in the field as well, as evidenced by a 30-day review of our IND. And finally, we've been able to get high levels of homologous recombination. In addition to the investments in the platform, we make significant, we use the same kind of thoughtful approach to business development. We partnered with Allergan in the area of ophthalmology. They're an expert in that area. We partnered originally with Juno, one of the 2 or 3 original CAR T companies. They've now been acquired by Celgene, and they obviously remain committed to that area. Last year, we won the interference, which says that the patent that we have that allows CRISPR-Cas9 editing in eukaryotic cells as a therapeutic reaffirms our ownership of that patent. That'll be an important piece of the IP for anybody who wants to make CRISPR-Cas9 medicines at all. And so with that, let me just reiterate the key messages. We're now a clinical stage company with LCA10. The progress we've made there sets up the S2A program and eventually other in vivo editing approaches. We've made good progress on the sickle cell program. That's going to be our first entry, and so this year we anticipate IND-enabling activities for USH2A and IND-enabling activities for the sickle cell program. Those will be the next 2 programs you'll hear going into the clinic. And we're very active in building an oncology business, both with engineered T cells and with NK cells, among other things. And with that, I'll pause and be happy to take any questions for the remaining few minutes we're here.
As we look forward to dosing the first patients in the LCA trial, how would you characterize the risk to success in that trial? What are the key factors that you think will determine whether you can see a clinical impact of gene editing?
Do I need to repeat your question, or do other people? Okay. The question was, what are the critical factors necessary to see benefit in the LCA10? I think that we're poised in a good way with the LCA10 program. We really understand the biology. We're going right at correcting the molecular defect, and we have a tremendous body of pharmacology and safety data that puts us in a good position to be confident about the dose selection. We will obviously start in patients where the risk is lower, and therefore the potential for benefit is also lower. And so I think as we get into patients with some vision, some of the risks that go with whether the cortical pathways or visual pathways are still intact is probably the biggest risk with the original set of patients, since it's not clear that those visual pathways that go from sensation of light to construction of a vision, it's unclear how intact they are in patients that have never had vision.
Are you going to be using the CPF1 enzyme too, or is it, I know there is another company, Croston, in being collaborated. So, are you going to be using the CPF1 in your pipeline?
The question was, are we going to be using CPF1 in our pipeline? The answer is, simply put, yes. And so you'll see more about that this year. It's a really good enzyme, number one. Intrinsically, CPF1 and Staph aureus Cas9, which we have unique access to, are more specific than Strep pyogenes Cas9. The Staph aureus Cas9 obviously lets us make the AAV that I just talked about. CPF1 allows us to make unique edits. And I think you'll see more about that as we talk about the sickle cell program later this year, where we're able to do some things that you cannot do with the Cas9. We haven't disclosed that yet, but stay tuned. So we've incorporated all the enzymes in our platform. And so much the way you think about screening a small molecule library, we always screen against all three enzymes.
Is there going to be any IP?
The CPF1 IP stands independent of the Cas9 IP, and we've solely licensed that from the Broad. So all of the conflicts that go with Cas9 do not apply to CPF1. Cas-X? I'm actually not familiar with Cas-X. Okay. Oh, is that the new enzyme out of Berkeley? Yeah. So that's a version, I think, of CPF1, at first blush, it looks quite similar to CPF1 to us. I didn't know the tricky name trade name they've.
So is because how's your technology different from CRISPR therapeutics? I know you put out some press release that is a better and similar to the same.
We're differentiated. I think to rephrase your question, how we differ than CRISPR. I don't want to go directly in commenting on competitors. Suffice it to say that we differentiate in two important ways. We're losing the lights there. That's okay. Is that a message? We have a differentiated platform. We have a differentiated portfolio, and let me say a couple words about that. On the platform side that I know you all gloss over when I go over, but as the person who runs a portfolio, it's like a big deal. We have more enzymes, so that we can do AAV delivery, and we have CPF1, which is going to turn out to be incredibly useful. And we've built this a robust suite of looking at on and off target editing rates, which are absolutely essential to be able to develop medicines in this field. And then we have a differentiated portfolio. So we have a reason to believe that our medicines are going to be differentiated. And that differentiated is based on the product, not the technology. So when I talk about differentiation, I don't mean we use a different technology than somebody else used to accomplish the same therapeutic goal. I mean, we think the products are going to be better. They may not all be better because these species don't always work out, but we have a reason to believe when we start a program that we're going to make something that's better than the things that are in the pipelines of other people's areas. So I'm not going to comment specifically on any one company, but those are the two things that we believe make us stand out from our competitors.
Are you going to be using CPF1 in the editing of in the cancer indication too?
There's a good chance you'll see CPF1 show up all over the place, and so it's a really good enzyme. And as we disclosed at Keystone last month, when you start to do multiplex editing, the combination of Cas9 and CPF1 dramatically decreases translocations. So it allows to do more edits with less translocations, which will, I think, can turn out to be incredibly important as you do multiplex editing for cell-based medicines.
So when I know when the news came out about CPF1 discovery by GAN, then one of the things mentioned was that it's better than Cas9 in neurodegenerative diseases, which is less side effects, and it is better in that. For example, Parkinson's, so do you think that are you going to be working on that?
We haven't disclosed what in vivo indications we're going after we do ophthalmology. Suffice it to say that it's an area of active discussion, actually active work in the company, but we haven't disclosed where we're going. And I think our AAV platform to deliver the editing machine really sets us up well to go anywhere that AAV can go right now. Great, thank you.